All right, this video is gonna go over quiz 1.5, which focuses mostly on the topic, which I will open up here, that asks you to think about the structure and function of biological molecules, which is a repeating theme in this unit. Um, just the relationship between structure and function, but more specifically to predict the causes or effects of a change in one of the components. So you're thinking about proteins. What happens if you change a type of amino acid or the number of amino acids? If you think of carbohydrates, what if you change the number of glucose monomers that are connected or the number of oxygen double bonds? Same for lipids. What if you increase or decrease the number of carbon-carbon double bonds in a lipid? How does that change the the function of the entire biological molecule? Images will be used to assess this, as well as written descriptions. So in our first one here, we have a protein, the CFTR protein. The name of the protein doesn't almost never really matters. It's kind of like Fred when it comes to a scientific name. You can just store it. It's the name of a protein, but don't let it throw you. It's made up of a certain number of amino acids in a chain. Some humans produce a version of the CFTR protein in which phenylalanine, which is an amino acid, has been deleted from position 508. And you're asked to think of which best predicts how the deletion will affect the structure of the protein. So we know if we remove one of the monomers, this is going to change the way that the protein folds. This question, when I look at the prompts, is clearly assessing that you understand the difference between primary, secondary, and tertiary structures in a protein. That is something you should be able to visualize happening. When the ribosome puts the protein or puts the amino acids together, it creates the primary structure of the protein. The secondary structure then is created by hydrogen bonding between the amino acids, which causes it to coil or fold into sheets. The tertiary structure then is created by hydrophobic and hydrophilic interactions of the R groups or the side chains. And sometimes it's other kinds of interactions. It'll form covalent bonds and things like that. So this will affect, if you change the number of amino acids you have, you take one away, it's going to change the way the whole protein is put together. So it definitely changes the primary structure. Excuse me. The primary structure determines the secondary and tertiary structure. So D is the correct answer. B was the one most of you chose that was the distractor. You correctly identified it does affect the primary structure, but you cannot change the basic shape without changing all of the other shapes above that. You change the basic structure, the primary structure, then all the other structures will be changed. Okay, question number 2. We have a cysteine amino acid, and they said that they find this amino acid in proteins found in organisms that live in harsh environments. And as soon as I hear that idea of harsh environments, I think of a place where it's difficult for proteins to stay folded correctly. That's usually what the problem is with places that are too hot or maybe too acidic. Environments that are usually too difficult for things to live in, the proteins often don't fold correctly. So they're saying in those kinds of environments, this amino acid is more common. Which of the following best describes the effect of a greater number of cysteine amino acids on the stability of proteins? Well, my guess is it's going to make them more stable, because that means that harsh environment is less likely to cause them to unfold or to change shape, and hence the animal or plant or organism will be able to continue to produce usable proteins. So I think it's B, which is the one most of you selected. I like that D was the distractor that most of you who got it wrong selected, because you're still thinking about increased stability. Now, it does say up here that bonds can form between sulfur atoms of different cysteine amino acids, but proteins don't fold up so that one amino acid connects to the one directly next to it by their R groups. Remember, the R groups aren't how the amino acids actually connect in the peptide bond. That happens in a different part of the amino acid structure. It's the R groups, the R group which is sticking off the central carbon here, that is actually interacting, right? This is where the sulfur is. The amine, or the peptide bond forms between the amine side of the amino acid, this NH2 here, and the carboxyl side, this COH with a double bonded oxygen, of a nearby amino acid. So those two don't have the sulfur in it, right? But there's a sulfur here in the R group, which means once you get that long primary structure, it can fold into some kind of crazy shape where those R groups are bonded together. And because sulfur forms these bonds with each other, not just an attraction but an actual chemical bond, they're going to more tightly connect to each other. So it's the tertiary structure of the protein that will be affected by the sulfur bonds, because the primary structure isn't changed by the R groups. Neither is the secondary. That's created by hydrogen bonding, but it's the tertiary structure. So really this question is assessing, can you differentiate primary, secondary, and tertiary structure and what leads to each? I would make flashcards or draw that process out until you feel comfortable with it. Okay. Third one, we have a mutated amino acid sequence that creates a protein. In the description, if you read about it, it says a mutation leads to a hydrophobic arm being deleted, and then it asks you to predict what will happen. Well, again, this is testing your understanding of the behavior of different amino acids, specifically the R groups, which can be hydrophobic or hydrophilic. And in a hydrophobic R group, or hydrophobic R groups are afraid of water, since proteins are usually found in a watery environment. The hydrophobic groups move into the center of the folded secondary structure of the protein. So that means these little black dots here are going to be in the center. Whether there's two sets of them, they'll get folded into the center, or whether there's only one, they'll get folded into the center. But that doesn't mean that they're going to behave exactly the same. So most of you selected the correct answer, right? And you showed the hydrophobic region in the center and again in the center. And these two proteins look basically the same. There's some little differences here and there, which would occur if those hydrophobic groups couldn't get into the center or weren't there at all. These two proteins the mutated protein will not function like the original protein. I know to you and I, they look pretty similar, but they're completely different and they won't behave the same way. So that mutation completely changed the function of this protein. Now, these two, I think this is testing the idea that you understand hydrophobic and hydrophilic. So if you got it wrong, if you got B wrong, you probably thought these would be sticking out, but then you were mixing up hydrophobic and hydrophilic. This is asking this is asking for the tertiary structure. This isn't a tertiary structure. This is a primary, or maybe a secondary structure with the folded sheets. And this just doesn't have anything to do with a protein. All right. I hope that makes sense. Please let me know if you have questions.